Hi everyone, my name is Eric Appel, and today I'm going to be talking a little bit about development of new technology for encapsulation and controlled delivery of monoclonal antibodies. So my lab in general is located in the Material Science and Engineering Department at Stanford University, and we focus on developing new materials for encapsulation and controlled delivery of active pharmaceutical ingredients, and we have a couple side projects on other fun stuff. But in general, we're focusing on getting active ingredients to where they need to be when they need to be there. And really, one of the things I'd like to focus on is that there's been such a huge shift in the pharmaceutical sciences over the last 15 or so years from small molecules to protein drugs. And you can see, even though this is relatively old, in 2004, all of the drugs, the top drugs were small molecules, but by 2014, that had almost switched entirely to protein therapeutics, and in particular, antibodies. And this is generally because proteins have very high specificity, high potency, high safety. But some of the disadvantages of these kinds of materials is, oftentimes they can have relatively poor stability and potential for immunogenicity. And while there's been a lot of work on protein engineering applications in order to, or approaches to engineer more potent materials, safer materials, more stable materials, there's also a major role to be played in developing new technology for formulation of these materials. And I like to highlight this quote oftentimes, which is highlighting that one of the major hurdles for the formulation scientists in developing protein formulations is the limited number of excipients commonly used in parenteral formulations. And this is often because the list of standard excipients was sort of grandfathered in from small molecule drug products. And oftentimes these compounds just simply don't address the primary needs for stabilization and controlled delivery of the large and complex protein molecules that make up this landscape. In particular, one of the, while there's a lot of depot technologies that have been developed for small molecules, such as PLGA microparticles or things like this, hydrogels have been one of the more promising platforms for encapsulating and delivering proteins, primarily because they are mostly made up of water. So hydrogels are water-swollen networks of polymers. The fact that they are heavily water-based means that any encapsulated cargo is maintained in their native aqueous environment, and so it can help control their or maintain their stability over time. But also by encapsulating cargo in this mesh, you can slow their diffusion and therefore slow their release and extend their release over a long period of time. Historically, that's been primarily controlled just by controlling the mesh size relative to the size of the drug, right? So small drugs relative to the pore size in the gels means that you get very fast release. By making the mesh size smaller, you can slow the release. But eventually, if the mesh size is too small in sort of a traditional hydrogel system, you can get complete immobilization of your cargo. This is somewhat like a soccer ball getting stuck in a soccer net, where you need to introduce some other mechanism to expand these pores to enable controlled delivery. So, but later on, I'm going to talk about another branch besides these, where rather than having the pores themselves open up, if you can have a system that is just simply dissolves away, the rate of dissolution can control your release of cargo. One other feature that I think is important to highlight is that while most of the work has been focused on developing chemically cross-linked hydrogels, these have some other drawbacks that are more pragmatic, in that it's challenging to get these kinds of materials into the body or to form them in situ in the body. And this is where an alternative approach is to leverage physically cross-linked systems. So by using strong and directional non-covalent interactions between polymer chains, you can actually build hydrogel materials that are easily formed and easily administered by injection. And you can see if you actually look at articles per year as a measure of interest in this field, while there's been an enormous growth in the interest in hydrogels generally, there's been a significantly higher rate of growth in injectable hydrogels because things need to be injectable in order to create useful drug products. And so our workhorse materials we call polymer nanoparticle hydrogels, and it's a simple but descriptive name because we have a polymer component, which is a dodecyl modified hydroxypropylmethylcellulose that we simply mix with biodegradable PEG PLA nanoparticles. And so this is what that process looks like, where if we take a solution of polymer and a solution of particle and just mix it together. In this case, we're doing it on a relatively small scale, but this is how we do it in the lab on a regular basis. This process is quick and easy, and we end up with a very nice homogeneous hydrogel system that's bubble-free, already loaded in a syringe, and so you can simply uncap, put a needle on, and administer. This process is mild. It doesn't require any additional heat or light or chemical reactions in order to occur. And so it's very simple to encapsulate cargo. We simply dissolve whatever our cargo is, typically with the nanoparticle solution, and then you end up with 100% of your cargo encapsulated in these gels. Also, because these materials can be so easily formed, it's really easy to scale their manufacturing. And so we've been able to show that here, using storage modulus as a metric of their mechanical properties, that no matter what scale you are making these materials on, whether it's a 0.5 ml in an Eppendorf tube or a 15 liter batch in a big bucket, the mechanical properties are identical across all scales, which is really critical for their translatability. These materials are very easily administered by injection as well. So they're very highly shear thinning. And so that's what this plot is showing. This is viscosity versus shear rate. And so while these materials have a very high viscosity under low shear rates, if you apply a stress, you see a very enormous drop. I mean, this is a 6 order of magnitude drop in viscosity of these materials, which allows them to be easily administered through standard needles. And here you can see in the video that immediately upon administration, the materials reform into a nice, solid, shape-persistent depot that you can envision retained or being retained in tissues and enabling long-term delivery in the body. So these materials we've been able to leverage to encapsulate a number of different cargo, and it with protein cargo, it has two important outcomes. One is stabilization of the cargo. So on the top here, I'm looking at two different experiments that we did where we encapsulated lysozyme and insulin and subjected these either encapsulated in a gel or in a standard saline buffer to stressed aging. So this was continuous agitation at 37 degrees. And you could see that in both cases, you have a major loss in potency here, or in this case, this is looking at aggregation of the insulin in standard formulations in very short order, but encapsulation in the gel dramatically prolongs the stability of the cargo. We've also been able to demonstrate that by simply tuning the formulation, so changing the polymer or the particle ratios or the total content of the hydrogel components, we can very widely tune the rate of release of the cargo. And so this is just looking at serum albumin in 3 different hydrogels having very different release profiles. And so this allows for fine tuning of the release characteristics of these materials. And so these gels we've been able to leverage in a number of different ways. So one case was leveraging the fact that these are can be easily sprayed and are very safe for prevention of postoperative adhesions. We've also leveraged this depot technology as a for encapsulation and controlled delivery of therapeutic cells. And so this was an example using human mesenchymal stem cells where we showed that encapsulation in this hydrogel depot could actually protect the cells from the host immune system and improve localized delivery. We've also used very similar kinds of materials for more durable retention of fire retardants on vegetation, actually to sort of create a vaccine-like treatment for high-risk landscapes from wildfire, which in California, this is a big deal currently. But today I want to focus mostly on our efforts to use these materials for encapsulation and slow delivery of potent neutralizing antibodies for infectious diseases. And so we've worked on a couple of different targets, and really from my perspective, it seems like there's a lot of potential in this space, both in the pre-exposure prophylaxis and post-exposure prophylaxis approaches. And so at first, we used just a simple model system where we used a mouse anti-ova antibody, and we could just simply encapsulate this in our gels, and we were able to show that with one particular gel formulation that I'm showing here, we had very long-term release, which we hypothesized would be enough to demonstrate a prolonged pharmacokinetics upon administration in an animal. We also showed that these gels can maintain the bioactivity of these materials. So similar to what we showed before, this is bioactivity here was just ELISA binding capability, that the overall ELISA binding was maintained over a long time during stressed aging, whereas standard formulations in buffer, the antibodies rather quickly lost their ability to bind their target. We first evaluated these materials in a simple mouse model. So we used C57 black 6 mice, and these again were mouse monoclonal antibodies. And you can see here that when dosed subcutaneously in standard saline and PBS, that we have a profile that looks pretty normal, where we have Cmax after 2 to 3 days, and then a elimination half-life of roughly 4 days, which is what I would expect from a mouse IgG. But upon encapsulation in the gel, we could see that we have both a reduction in Cmax and a prolongation of the pharmacokinetic profile. And so these dotted lines here are actually fitting this data using this simple pharmacokinetic modeling up here. But what this modeling allowed us to do is to gain some insight into the actual in vivo release behavior of the hydrogel depots. So we could get some idea of how quickly the materials are actually degrading and how quickly the cargo is being released. But overall, this was a very promising study because we saw the overall half-life of the pharmacokinetics extended roughly 5-fold from 4 to 20 days or so. But using the release characteristics, we were able to plug this into our simple model and use human pharmacokinetic parameters to try to characterize how we would expect that these gel materials might actually behave in a depot formulation in a human. And we could see that, again, this is a really promising extension of the pharmacokinetics from a standard saline subcutaneous bolus to a gel-based bolus, which can really prolong the time frame over which we can maintain a therapeutically relevant concentration. Here I'm just sort of arbitrarily picking 1 microgram per mil. But again, that was just a simple model, and so we first wanted to evaluate, or that we then wanted to evaluate with a proper monoclonal antibody, and so we started partnering with Distributed Bio and now Syntevax, because they had developed a potent SARS-CoV-2 antibody that was computationally engineered from known neutralizers of SARS-CoV-1. So this was fairly early in the pandemic that we were working with them on this. And we also used a more appropriate animal model, since this was a human monoclonal. So we used the humanized FcRn skid mice so that we could avoid anti-drug antibodies, because we need to be able to track the pharmacokinetics in these cases out to a long time frame. And so here we first evaluated IV bolus and subcutaneous bolus of these drugs, and we're able to get relevant pharmacokinetic profiles that would also help modeling in our gel case. So we then took these mAbs and encapsulated them in hydrogels and evaluated them in the same mouse model. We tested the same gel formulation, but with two different buffers to see if there was any difference in the buffers, and we could see that there wasn't. And overall, the hydrogels were able to significantly prolong the rate or the time frame of release of these mAbs in this animal model. We found that the bioavailability was really good, and in comparison to our subcutaneous bolus, and this is normalized by the IV dosing. And if we collected serum at day 7, when the overall titer of antibody in the animals in our two bolus groups as well as our two gel groups was roughly similar, we could see that the potency in a pseudoviria neutralization assay that we conducted in with Peter Kim here at Stanford demonstrated that overall these hydrogels, this hydrogel depot technology, can prolong the pharmacokinetics of these monoclonal antibodies and maintain their potency upon administration. So this was all very promising, and we have future work expanding to other mAbs, including several HIV mAbs, and we're looking forward to getting some more of that data soon. In the final part of my talk, I wanted to just switch gears and rather than talking about addressing the pharmacokinetic challenges associated with some proteins, talk again about their stability and describe a new excipient technology that we've developed that can dramatically improve the stability of protein drugs by preventing adsorption of those drugs to interfaces, which is one of the primary targets of aggregation. And so initially we started working with insulin just because this was a simple protein that we could acquire in our lab relatively easily. We used a high throughput combinatorial synthesis approach to create a library of roughly 800 unique combinatorial copolymers made of hydrophilic monomers as well as some sort of functional dopant monomers. And then we were able to screen them for their ability to stabilize insulin in a stress staging assay. And so here what I'm showing is time to aggregation in an assay where we're just looking at a change in transmittance to measure the actual formation of amyloid fibrils in the insulin. And so while insulin in this assay aggregates in roughly 12 hours or so, if you add pluronic to the formulation, which is the primary polymer excipient that's actually used in commercial insulin formulations, you get a roughly twofold extension in the time to aggregation in these formulations. But as you can see here, we had a number of hits where the time to aggregation was extended all the way out to the 30-day endpoint of these studies. And so we had a number of hit copolymers that extended stability roughly 55-fold over standard commercial insulin formulations. We then tried to evaluate in these insulin formulations our new polymer, which we call Moni. So this was our lead candidate, where we simply added it to vials of commercial Humulin, which is a Eli Lilly product of recombinant human insulin. And we evaluated this over the course of 6 months of continuous stress staging. And so here we're showing formulation age and aggregation. You can see that Humulin aggregates in just under 2 weeks, whereas our formulation was completely stable throughout this entire period of stress staging. Using circular dichroism as well as an in vitro assay of bioactivity, we were able to show that the insulin was completely stabilized throughout this entire 6-month period. We also evaluated these formulations in a rat model of insulin deficient diabetes to show the actual function. And we were able to show again, after 2, 4, and 6 months of continuous stress staging, the pharmacokinetic and pharmacodynamics of these formulations were completely maintained throughout this stress staging experiment. And so what we wanted to do is to try to leverage this technology and see if it could also be used to stabilize proteins in formulation. And so we started working with PGT121, which is an anti-HIV antibody, and we were able to first just sort of evaluate the mechanism of action, demonstrating that our Moni polymer really does preclude PGT121 adsorption to interfaces, using both surface tension measurements and interfacial rheology measurements. Here, this is just showing that in a standard formulation, you have PGT121 adsorbing to an interface, and this actually forms a viscoelastic gel-like aggregated structure at the interface that you can actually measure on a rheometer. Whereas as soon as Moni is added here, this is at the detection limit of the instrument, and so we completely prevent the formation of this viscoelastic layer. And this dramatically stabilizes the cargo to stressed aging. And so this is looking at monomer content in formulations that were continuously agitated at 50 Celsius. And so in a standard formulation, the PGT121 goes bad relatively quickly, but in the presence of Moni, it's stabilized for over 3 weeks. We also demonstrated in the same SCID mouse model of with human FcRn that the overall pharmacokinetics is completely maintained upon administration alongside this Moni polymer. And so whether we dosed a low concentration IV or a very high concentration of MAB stabilized by our Moni polymer, we could maintain the pharmacokinetic profiles and overall the area under the curve as well. And so this was just really promising, demonstrating that overall this Moni polymer has some very unique capabilities to stabilize protein cargo in formulation. It's really unprecedented in its ability to stabilize these proteins, and it has numerous benefits that make it a promising candidate for as a new excipient for protein formulations. So I would love to acknowledge our collaborators, Peter Kim and Abby Powell, who was a postdoc in the Kim lab, Jake Glanville from Distributed Bio for working with us on the SARS-CoV-2 antibody, and Mike Farzan, who we've been working with on a number of other projects. The two students in my lab that did all the work were Anthony Yu, one of my very first graduate students, and Katie Meese, who's about to graduate. And I'd like to thank both the NIAID and the Bill and Melinda Gates Foundation for funding this research. Thank you very much.